that I think there's been a lot more emphasis on team science. Experimental immunology takes incredible training, and it's hard to expect that someone who is able to do very intricate experiments, for example mouse models of multiple sclerosis, is also going to know what to do with a high dimensional data set. And so there are all of these partnerships that have been developing and that I've been a part of, and I see just across the field. And so I'm very excited about this team approach to science, combining experimentalists and computational folks. I'm also very excited about breakthroughs in computational biology and machine learning that enable us to take many data sets. So you could have these induced pluripotent stem cells from different patients, but of course, even if you are able to generate an intestine in a dish, it's missing some key things. What about the nervous system? What about blood flow? So it's not a perfect system. In parallel, there are a lot of important studies in model organisms, for example, in mice, where you do have access to a real intestine from a real mouse, and you would never have access to something like this in humans. And so some techniques are available from machine learning, for example, multitask learning approaches, where you might be able to reverse engineer, say, how a human organoid responds to a disease, but to take into account how a mouse organ also responds to that same disease, and to learn both of those models together. And so that's an exciting direction. But I think the thing that has struck me is that now it's actually possible to measure all of the genes that are expressed in a cell. Through proteomics, you can measure a wide variety of the proteins and the post-translational modifications. You can measure the metabolome, a lot of metabolic metabolites that are in a cell, the state of the DNA, so whether parts of the genome are being used to regulate gene expression. So you have all these opportunities to make measurements. And across the board, because we can, people are wanting to look at more than, say, 5 variables when they're looking at a particular cell type, and want to use genomics technologies. And so I think the way things have changed the most is that there's a wonderful opportunity for computational biologists and people with computer science backgrounds to have an impact on experimental immunology.